Can others should join? Wait a little bit. Okay. Right now, what is the difference between PS1 and PS2? If we look the representative picture of photo system, this is the picture. Only the difference is if we look PS1 and PS2, they have a different composition, but the function is to absorb the light. Photosystem 1 absorb the light. Photosystem 2 can also absorb light. The difference is there are pigments and there are other proteins. That is the composition. When the composition different, the amount of light absorbed by PS1 and PS2 are different. Okay, next we have to learn a little bit about the light because light brings the energy. Right, where we get the light? We get the light from sun. So what brings us the light? So it brings us the energy. Okay, now there is a nuclear reaction take place in the sun. So this nuclear reaction produce a lot of radiation energy. So this energy produced by the sun can reach Earth through a radiation, right? Because if we look at the heat as an energy, if heat needs to come to Earth, it can come through the air, it can come through the solid surface, it can come through the vacuum. Now space, we don't have air, we don't have a solid surface, it is more vacuum. So then it's coming from radiation. So this energy come as a electromagnetic radiation as a waves. So the powerful is the gamma rays, they have a short wavelength. Then the X-rays, they again have a short wavelength. Then next is a UV light. Then the visible light, then infrared light and radio waves. Gamma rays, X-rays, UV, visible, infrared, radio waves. So electromagnetic radiations emit from the sun, release these kind of rays. But when it come to Earth, we get mostly UV, visible, and infrared. UV light, visible light, and infrared. And out of these three, we get mostly visible light and infrared light. So infrared light used to heat the Earth's surface, and visible light is used mostly by photosynthesis, because this has energy. UV light has energy, visible light has energy, and infrared light has energy. So plants are specialized to take the visible light. So visible light wavelength starting from 400 nanometer to 700 nanometer. And in this nanometer, the first 400 nanometer is violet, then we call it indigo, a blue. So it's more blue. It's more related to blue color, and in the middle is green color. Then yellow, orange, and red. So these are the 7 colors of rainbow, 7 colors of the light: violet, indigo, blue, green, yellow, orange, and red. And from this, we consider this as a red color, and this is a violet and blue. So violet, so it's a violet and blue color, and this is a red color. Why I just separated now violet and blue into one color, red into one color? Because we don't consider in the resource book and the textbook each like violet, indigo, blue, green, yellow, orange, red as a separately. So we separate the one end 400 nanometer end like a blue violet range, and the 700 nanometer range, it's a red. So this area is a more red color. So then we can separate into two colors, the whole visible spectrum. 400 nanometer in violet blue, and the 700 nanometer red color. And also we have a third color now, that is a green color. This one. So 7 colors we can summarize into 3 colors: violet, blue, in the middle is green, other side is red. Okay, now why it is important to understand this light? Because light brings us the energy. Key light capturing pigment is chlorophyll A. Other pigments, chlorophyll B and carotenoids. Okay, then what is the function of chlorophyll A? It can absorb light and involve in light dependent reaction. So I can write here 2 functions. Chlorophyll A can absorb light, involve in light dependent reactions. So in this case, the highest. The highest light absorption done by the chlorophyll A. Chlorophyll A involved directly in light dependent reactions. So its role is very important in light dependent reaction. As a result of that, chlorophyll A is the main key light capturing pigment. Chlorophyll B, it absorbs light. Carotenoid, it also absorbs light. Carotenoid also has another function, photoprotection. So chlorophyll has 2 functions. Chlorophyll A has 2 functions. Chlorophyll B has 1 function. Chlorophyll carotenoid has again 2 functions: absorb light and photoprotection. Okay, light absorption we can understand. All pigments can absorb light, and light dependent reaction, its role, we haven't learned it yet, so we can't explain. What is photoprotection? So it gives some idea, protection from the light. Photoprotection is protection from the light. So we'll see why it is important. Now this word is used, dissipation, in the resource book. So dissipation is if there is too much energy, this energy is distributed to different areas, then there is no harm from the heat. So you need to remember this word because it's a technical term: absorption and dissipation of excessive light energy by carotenoid. Okay, this is again very important thing. Oh. Absorption and dissipation of excessive light by carotenoids. Okay, why is that? Because if the photosystem absorbs light, Then this light is moving down to the reaction center. So middle is the reaction center. So all light captured by the light harvesting complex finally move into the reaction center. So when the light is absorbing, it brings to the reaction center by distributing everywhere, not as a very strong spot of the light. So this has two effects, right? So photoprotection prevent damage from light. So we can say carotenoid are important in photoprotection, and it prevent from damaging from the sunlight. Second thing, prevent formation of reactive oxygen molecules. Protect formation of reactive oxygen molecules. Probably this word reactive oxygen molecules is also written as reactive oxygen species, right? So you can also remember this reactive oxygen species. It's the same thing, reactive oxygen species and reactive oxygen molecules. Okay, what is the function of carotenoids? It can absorb light and it contribute to the photosynthesis. Second function is it does photoprotection. What is photoprotection? One, it prevents damaging from excessive sunlight by dissipation of excessive sunlight. Second, it prevents formation of reactive oxygen species. Okay, now reactive oxygen molecules we don't learn uh, in the syllabus. Okay, now I am going to ask you a question. What is the function of peroxisome? So we have learned this. What is the function of peroxisome? If you want to get a really high marks with a really big level A, you can't forget the fast because this exam depend on the things which you can remember all the time. What we have done, like in the past we have done the lessons, and when it comes to today you can remember all the lessons, right? So if at this point you can remember everything. Metabolism is very general. Apurva, it's a general term. Metabolism is catabolism and anabolism. It's a reactions, making molecules, breaking molecules. If you can specify what kind of metabolism It's fine because it is metabolism is reactions. So everything what we learn are chemical reactions. Peroxisomes are involved in what kind of reactions? Lipid metabolism. At least you are a little bit closer. There is a similar structure. You can check the notes called glyoxysome. This we can find in the plants and animals. Glyoxysome we can find in the plants. Glyoxysome function is it convert fatty acid into carbohydrate. So this is the function of glyoxysome. The peroxisome has two functions. That's very important, two functions. So if you trying to pass this exam to get a big marks, what we have learned is this white line. So all these things you should remember today, because if you are waiting to the exam, you are building up so much of things which you can't finish. You can't finish these things. Reason is uh, this exam, if you want to pass and get a good marks, the commitments, you have to keep the commitment from the first day to the end of the exam, because most of the students when they finish the O level, next day they are highly motivated to start the A level class. So they are looking A level class, and maybe after a few days they join the A level class. After that, the student overall interest and motivation is like this. But with the time, it is decreasing. Now, even these days, so 2023 paper passing කරගන්න බැරිද? So 22 may now there is no such a way that you can do miracle at the end. The student trying to do something differently at the end, at least 6 months before the exam, or at least 1 year before the exam, then the bus is gone. So this exam, you can pass the exam if you have the motivation from the first day to the last paper of the exam. Otherwise, it is very difficult. So we should understand this is you are trying to sit for the difficult exam. Biology, why biology is difficult? Because the contents are high. You have to remember too much contents. I think best way to make it easy, make it to happen. Uh, don't wait until you are coming to a certain point. So take again. I can tell you something that if you can do it like just download this resource book to the mobile phone instead of checking this TikTok, this thing, Instagram and the Facebook like thing. You can look whenever you are traveling or free. You can start reading the book. That's actually help to revise quickly, because otherwise things are forgetting. Okay, so this has two function. One is it can neutralize peroxidase. Sorry, detoxify hydrogen peroxide. Second one, It is important in photorespiration. Okay, now photorespiration we haven't learned. You don't know what is photorespiration. But if you go back the notes, it is written peroxisome has one function of photorespiration. So you don't know what is photorespiration. And you don't know what is this effect of hydrogen peroxide, where it comes and why these hydrogen peroxides are important like things, right? Uh, right, go back here. What is the function of carotenoids? Two functions: prevent damage from light, prevent formation of reactive oxygen molecules. We don't learn reactive oxygen molecules in the syllabus, but we just use the word reactive oxygen molecules. And here we learn detoxification of hydrogen peroxide. These kind of a thing you need to remember well. But for understanding, we can also check this. I can also explain like this way, right? What is it? If too much light, then this oxygen absorb the light energy, and oxygen can remove its electron. So electron can remove after oxygen remove its electron. After oxygen lost its electrons, the oxygen is a little bit of reactive. So this molecule is very reactive because it lost electrons, and the molecule is not stable. And only thing cell can do this can convert into a hydrogen peroxide. So this hydrogen peroxide can neutralize into water and oxygen by enzyme catalase. Now you have learned why hydrogen peroxides are produced and why it is important to detoxify hydrogen peroxide. Because this hydrogen peroxide can also convert into another reactive oxygen molecules like OH, like this. So these molecules can damage the organelles, membranes, DNA like things. If you want to see this, we can buy hydrogen peroxide from the pharmacy, and if you add piece of potato, you see bubbles are coming. So take hydrogen peroxide into tube and just add piece of potatoes, because potato has catalase. So catalase neutralize hydrogen peroxide by forming water and oxygen. Okay, this is something extra knowledge just for the interest to explain reactive oxygen molecules. So you can remember reactive oxygen molecules are harmful. They can damage the cell. Excessive light can form reactive oxygen molecules like this. Light, oxygen molecule, light energy is absorbed by the oxygen and electrons are released, because too much energy can excite the molecules to release electrons. So this is important thing to remember. The carotenoids functions is photoprotection, Okay, then we also learn absorption spectrum. So absorption spectrum is like this. In a graph, we have a two axes. One is y-axis, other one is x-axis. So x-axis, I place the wavelength. Wavelength visible light has 400 to 700. So y-axis, I place absorption. Uh, let's say percentage amount of light absorbed. Now you can understand like this. Uh, if I write here for 470, example here, 470, then I look what is the amount of light absorbed by the pigment. So if I take chlorophyll A and I check at 470 what is the light absorbed, then I can find, okay, I can mark here this much. 470, let's see this one like a 70%. So if the wavelength is 470, chlorophyll A can absorb 70% of light. Then again, I am trying to make another experiment. What is the absorption at 480, 490, 500? Likewise, I am measuring using a method what is the amount of light absorbed by each pigment. Then I plot the graph for chlorophyll A. So if I do this one, chlorophyll A is like this. Then I'm trying to draw the graph for the chlorophyll B. So chlorophyll B is like this. And also I'm doing now for carotenoid. Now if you look at this, graph, we can define absorption spectrum. If I want to define absorption spectrum in a graph, usually the definition is coming what is the relationship between x-axis and y-axis. What is the relationship? I can say amount of light absorbed at different wavelength. Its absorption spectrum is the amount of light absorbed at different wavelength. We can remember things by repeating many times, but you can also understand. If you understand it, you can also remember. Absorption spectrum is amount of light absorbed at different wavelengths. So different wavelengths is starting from 400 to 700, 401, 402, like that. And if I go back, to say what is the function of chlorophyll A, I said chlorophyll A absorb light highest. Why I call highest? Because if we look at this the spectrum, electromagnetic ray spectrum, the gamma rays are very powerful. They are very powerful. Then the X-rays are very powerful. Then the UV light is very powerful. Like that because of the wavelength. So they have a shorter wavelength and they are powerful. This area, the wavelength is longer. Now, if we take this visible light, 400 nanometer has more powerful energy than 700 nanometer because energy level is highest toward this side. It is highest toward this side, and energy is lower at this side because the radio waves like this. Radio waves they have less energy. So 400 nanometer is higher than 700 nanometer. So now you can understand why chlorophyll A can absorb higher light because it is more towards the 400. So here the light energy is more toward the 400, at least this part. And here also they are higher. So this explains chlorophyll A can absorb highest light. Then we are coming to the next action spectrum. So action spectrum is if I replace absorption with the rate of respiration, rate of photosynthesis. Absorption is replaced with the rate of photosynthesis. You can understand more light, more photosynthesis. So if more light, more photosynthesis, then action spectrum is something like this. It is similar to the absorption spectrum. So how we define action spectrum? Again, relationship between x and y axis. A rate of photosynthesis at different wavelength. So action spectrum is it's a rate of photosynthesis at different wavelength. Because you need to remember what is action spectrum. You need to remember what is absorption spectrum. Okay, we have finished that section. Key points to remember. Chlorophyll absorb violet, blue and red light. Chlorophyll A is the main or key light capturing pigments. Pigments form photosystems which locate in the thylakoid membrane. Carotenoid absorb and dissipate excessive light. That is called photoprotection. Now this is the summary of the lesson I explained to you. So you trying to remember from the beginning important things, definition, and who can do photosynthesis, why it is very important globally, and what are the two type of reactions, what are two type of plants, and the structure of chloroplast, and photosystems, and pigments, and the light energy. Okay, do you have any questions about this section so far? Okay, we have to join again for the next session to continue. And meantime, example of exam question. This is one example of exam MCQ question taken from recent exam.